Hello, this is Dr. Armand again, bringing you another pre-lab video lecture. And in this lecture, we'll be looking at experiment 2, observations of and reactions of household chemicals. So this experiment is divided in 2 parts. In the first part, we'll be looking at reacting different types of household chemicals with various reagents. And in the second part, we'll be looking at the reactivity of baking soda. So sit back, relax, and enjoy the show. So the first part, we're looking at observing the different types of chemical reactions with the household chemicals. And so before we begin actually talking about that, I wanted to just briefly discuss the types of chemical reactions that we can observe. And hopefully this will uh, help you write down or analyze the results of why certain reactions, certain household chemicals react and certain ones do not react. And so in the previous experiment 1, we looked at different signs to show that a reaction had occurred or has occurred. And one of those signs was a color change. So maybe when you mix two solutions together, we formed a solid. Another sign that a reaction has occurred is the evolution of gas. And we talked about the different types of ions, and when reacted with acids, they give off certain types of gases. And also we delved into the splint test, how we can determine the identity or determine the difference between hydrogen, oxygen, and carbon dioxide gas using the splint test. Another way is the absorption or evolution of heat. We refer to this as exo or endothermic. And the formation of an insoluble solid. So these are signs that a chemical reaction is occurring. Now we're going to talk about what types of chemical reactions we can have. And so chemical reactions are divided into 2 broad groups, which you'll learn about when if you're taking the lecture course. But these 2 broad groups are called redox reactions, as shown here. And the other type of category of chemical reactions are double displacement reactions. And these 2 are separate categories of reactions. You can't cross redox reactions with double displacement reactions. And so there are 4 common types of redox reactions, and we're just going to talk briefly about each one and the characteristics of each. Now, redox reactions involve the transfer of electrons. And you'll learn about this in your lecture course, but just to give you a very simplistic explanation of this. So let's say we have iron solid plus O2 gas gives Fe2O3. So here, this is a redox reaction because electrons have been transferred. And the way that you'll know that the electrons have been transferred is looking at what's called the oxidation number, or sometimes you can use it the same as the charge. And so for elements, the oxidation number is 0. And then for the elements in the compound, they would be non-zero. So this would be, for example, +3, and oxygen would be -2. And so since there's a change in the oxidation number of the different elements, then we have electrons being transferred. And redox reactions are used in a variety of different applications, especially batteries. Batteries are redox reactions. So the first type of redox reaction, a combustion reaction. And combustion reactions are very important because those are the reactions that help run the common conventional combustion motor. And so what you need for combustion is, of course, you need a fuel. Now, most of the time, the fuel is a hydrocarbon, and a hydrocarbon is a compound that contains carbon and oxygen. So you do need a fuel. It doesn't have to always be a hydrocarbon, and we'll give you an example of one that is not. And then it always reacts with oxygen gas. So you have to have oxygen gas for it to combust. So you need a fuel, which is oxygen, a fuel, which in this case is a hydrocarbon. You need it to react with oxygen gas, and you produce oxides of the fuel. So you're producing oxides of the fuel. So our fuel contains carbon and hydrogen. So our oxides would be carbon dioxide and water. So anytime you use a hydrocarbon and you combust it, you're always going to produce CO2 and water, and you produce a lot of energy as well. That's why your internal combustion motor gets hot when you're driving down the road, because you're doing this combustion reaction. It releases heat. The heat heats up your engine. That's why you have a radiator to dissipate the heat from the engine block. So it's a very exothermic, and it produces gas, because that's why you have an exhaust pipe to remove the gas from the combustion center. So this is one type of chemical reaction. Now, an example of this combustion reaction, not using a hydrocarbon, sometimes we can use hydrazine, which is a nitrogen containing compound. We can combust it, and it's also used as a rocket fuel, and we get oxides of the fuel. So here we use hydrazine. It's our fuel. We react it with oxygen. We get oxides of the fuel. So here we have nitrogen and hydrogen. We get nitrogen monoxide and steam in this example. So this is one type of redox reaction. The next type is called, or referred to as, synthesis. Now, in some books, synthesis you may also see the name combination. So it's the same thing, combination or synthesis reaction. The key characteristics of a synthesis or combination reaction is you have two or more reactants give you one product. So we have multiple reactants gives us one product. Now, there are different types of synthesis reactions. I'll just explain a few, but not all synthesis reactions are redox reactions. And so I'll give you one type of synthesis reaction, which is a redox reaction, and one example of a synthesis reaction that is most of the time not redox reaction. And so the one type of synthesis reaction that is a redox reaction is when you have an element, and there can be more than two elements. I'm just using two elements as a reference point. So you have an element plus an element gives us a compound. Now, anytime you have an element on one side, you have a compound on the other side, or the element is in a compound on the other side, redox has occurred. So this is always redox. And so the example for this is the one right above it. Iron plus sulfur gives us iron 2 sulfide. Now, another example, and this is about not all the different combinations, but just to show you the difference. You can have a compound plus compound. And again, I'm just using two reactants, but you can have more than two compounds. You can have 3 compounds, 4 compounds. Gives us a compound. Now, this is typically not redox. However, you may need to check the oxidation numbers of your reactants and products to make sure redox has not occurred, but usually it's not. And an example of this For example, barium oxide plus CO2. When you heat this up, or no, not heat it up, we form barium carbonate. So this is an example of a synthesis reaction that is not redox. So with synthesis combination, the key is 2 or more reactants, and you only form one product. The next one is decomposition. Decomposition is like the sister of synthesis. So here, the key thing for decomposition is that you have one reactant. So on the left-hand side, there's only one reactant, and you form 2 or more products. Now, as you see from the asterisk, this is sometimes may not be redox for the same reason as for synthesis. I'll show you one category, one combination, or one type of reaction that is always redox, one type that is not, but this is not by any means all the different combinations of a decomposition reaction. So for example, here we have a compound decomposes into its elements. So this is redox. This is always redox because, again, you have a compound on one side, they get put into elements on the other. And this example here that we show you is an example of that. So we have water decomposes into elements. Another example of a decomposition reaction is we take calcium carbonate, we get calcium oxide and CO2 gas. Now this is not redox. Oh wait, excuse me, sorry. So the other combination is a compound that decomposes into compounds. So an example of that, and this is usually not redox. And the reason I use usually is because there may be some oddball, or there may be a reaction out there that is compound decomposes into compounds, maybe redox, but usually it's not redox, but you can always check the oxidation numbers to see if redox has occurred or not. And the example here is the decomposition of calcium carbonate. So here it's a decomposition reaction because we have one reactant gives multiple products, but it's not redox because there's no change in the oxidation numbers. And then the last type of redox reaction is a single replacement reaction. And we talked about this in the previous lecture, but in a single replacement reaction, the more active element that likes to be paired up. So an element will replace another element in the compound as long as the elements, the reactant in the element form is more active than the one of the species in the compound on the reactant side, then redox will occur. So for example, we have magnesium and we have hydrogen. Magnesium is highly active, so it's going to come in and kick off the hydrogen, and we're going to displace hydrogen gas from water. Another example could be, we take some potassium, react it with zinc nitrate solution. Since potassium is more active than zinc, a reaction occurs, and we displace the zinc. And these are always redox, because always on one side you're going to have an element, on the other side it's going to be in a compound. And the way we know that redox, or which element is more active than the other, is something we call the activity series. It's a chart that shows you a list of the activity of different elements. So these are the different types of redox reactions. Now we're going to shift gears and talk about double displacement reactions. So with double displacement reactions, there are two subgroups of double displacement reactions. These two subgroups are precipitation reactions and acid-base reactions. Now, double displacement, sometimes you may see the term metathesis. So a metathesis reaction is a double displacement reaction. And so the first type of reaction, double displacement reaction we're going to talk about is precipitation reaction. Now, double displacement reactions are probably more about how you're going to explain why certain household chemicals react with others. You're going to be talking about, most of the time, double displacement reactions. So with precipitation reactions, the key thing is that you have two reactants that are solutions. So your reactants are solutions. When you mix them, you form at least one precipitate, which we call insoluble solid. So again, on the left-hand side, you have two aqueous solutions. What happens is the cations in each of these reactants switch positions. They displace one another. And we get two new products, and at least one of the products is a precipitate. So we take lead nitrate and potassium iodide. Lead nitrates and potassium switch positions. We get lead 2 iodide and potassium nitrate. Now you will use what's called the solubility rules to help you identify if lead 2 iodide or potassium nitrate is soluble. So potassium nitrate is soluble, so it gets the subscript or the heading or the label aqueous. Now lead 2 iodide is actually insoluble, so it should get the labeling of a solid. So when you take lead nitrate, mix it with potassium iodide, you're going to form lead 2 iodide. It's going to precipitate out, and potassium nitrate is going to remain in solution. So again, the two cations switch positions, we form two new products. Then we check the solubility rules to see which one of these products is soluble, which one is not. And then we can give the labeling of S for solid, meaning it's insoluble, and AQ for aqueous, meaning it's soluble. And you should be able to describe the precipitate. It's the insoluble solid. So for example, is it finally divided? Does it coagulate? Does it suspend in solution? Does it settle out? So these are some of the characteristics or key words you should use in describing your observations. Also, is it flocculent? Forms like cotton balls, kind of fluffy looking, or is it gelatinous, etc. And these are some examples of precipitate. So here on the right hand side, go back. So this would be, kind of flocculent here. It's suspended. It looks kind of fluffy. Here we have, a very maybe gelatinous looking precipitate. So you should be able to describe the precipitate that you observe. And just some other examples of types of precipitate. So again, double displacement reactions, two solutions mix, form at least one insoluble product. And so the way we determine which products are insoluble are the solubility rules. Now for at least this experiment, when you're doing questions involving solubility, make sure you refer to this slide here, because different solubility charts have slight differences depending on what they define as those ions that are moderately soluble. But just a few key things about solubility rules here. So the cations in the middle column, these are the soluble cations. The cations that are shown at the far right, these are the insoluble cations. And just a few key points here. As you see, all nitrates are soluble. So that's a very important thing to remember. Another common one is the halides, so chloride, bromide, and iodide. These halides, only these four cations are insoluble. It's another important one. So for example, silver chloride or lead iodide that we saw on the previous slide, these would be insoluble. Another one is sulfates. For sulfates, most cations are soluble except the ones listed here. So for example, barium sulfate, strontium sulfate, lead sulfate, lead-2 sulfate, these would be insoluble sulfate. So if your product is one of those, it would precipitate out. And then just one other one to kind of pick on, hydroxide. So most hydroxides are insoluble except ammonium group 1 hydroxides and your heavier group 2 hydroxides. So all other hydroxides are insoluble. So you use the solubility rules once you know the products of your double displacement reaction to identify which product, if any, are insoluble. And the last type of double displacement reaction is an acid-base reaction. So in an acid-base reaction, you always form a salt and water. And most of the time, a good way to know if you have an acid, most of the time, not always, by no means, but most of the time, hydrogen is listed first in the formula of the compound. So here we have some generic acid reacts with a base. So the cation of the base and the hydrogen of the acid displace one another, and we form a salt and water. And so the salt is always going to be the cation of the base and the anion of the acid. And these are the 6 common strong acids. Is probably a good thing to go ahead and commit these to memory. And so, notice in all of these, hydrogen is the first element listed in the formula. And that's at least for the majority of the time, that's a good way to know if you have an acid. Of course, the exception is water. Water has hydrogen listed first, but for at least Gen Chem 1 purposes, it's not an acid. And so it's very good for you to commit these 6 common strong acids to memory. Now, strong bases are going to be your soluble hydroxides. So if you remember on the previous slide, we said soluble hydroxides were group 1 and heavier group 2 hydroxides. So whenever you mix an acid and a base, you're going to produce a salt and water. Now, the one exception to this is if you mix an acid with a carbonate, as you found out in lab 1, when you mix an acid with a carbonate, in addition to producing salt and water, you produce carbon dioxide as well. So now we're going to show you the kind of the process in performing part 1 of the experiment. So in part 1, we'll be using the household chemicals shown here. So we'll be using So in part 1, we'll be using the following household chemicals. We'll be using baking soda, Lysol toilet bowl cleaner, bleach, and the toilet bowl, another type of Vanish bowl cleaner. And so all these have certain ingredients, certain key ingredients in them that we're going to look at. And so the Clorox bleach, we're going to be looking at the key ingredients. The 5%, it's made up of 5% sodium hypochlorite. Lysol toilet bowl cleaner, key ingredients 9.5% hydrochloric acid. Of course, baking soda is sodium bicarbonate. The crystal bowl cleaner, the key ingredient is 62% sodium bisulfate. And of course, we're going to be looking at blue food coloring as well. So these are the different types of household chemicals we'll be using. And here are the reagents we'll be using to react with the household chemicals. So we're going to be using silver nitrate solution, calcium chloride solution, hydrochloric acid, blue food coloring. We're going to react with the household chemicals, green food coloring, and Thymol Blue acid base indicator. Now, Thymol Blue is an acid base indicator. So if your solution is acidic, it's going to appear as one type of color. If your solution is basic, of course, it'll be a different type of color. So an acid base indicator has a unique color depending on if it's in an acidic solution or if it's in a basic solution. So now what you see here is we're using cell wall plates. So on the left-hand side, we have our reagents. At the top, we have our household chemicals. So we set it up as so, and these are the household chemicals you'll be using: Lysol, baking soda, Vanish, and bleach. But for this experiment, just to show you how we do it, we'll be using Drano as our example. And Drano, one of the key ingredients in Drano is sodium hydroxide. It's very caustic, very basic solution. So when we're doing our demonstrations, we're going to be using Drano, and I kind of explain some of the results that we get from Drano to help you analyze the results you get from your household chemicals. And so what we've done is we mixed a little bit of the Drano with water so it's not fully concentrated. So we take some of the Drano, mix it with water, we mix it up so that it is not as concentrated straight from the bottle. Now with the Lysol, When you're in the lab working with the Lysol, you're going to take about 1 milliliter of it and add it to 5 milliliters of water so that it's not so concentrated. So you'll take your 1 milliliter of Lysol, add it to the 5 milliliters of DI water, and mix it up. So Lysol is a blue solution, and it gets kind of soapy as well, Lysol. So we're mixing the water with the Lysol. And this is what we're going to use when we test the reactivity of Lysol, this dilute solution. So Lysol, that's how we prepare the solution. Next, we're going to prepare a solution of sodium bicarbonate. So we're going to weigh out approximately 0.3 grams of sodium bicarbonate and add it to about 10 milliliters of water. So using a 50 ml beaker, we're going to add the 10 milliliters of water and the sodium bicarbonate, stir it up, and get our solution. So again, we used about 0.3 grams of sodium bicarbonate. And you keep on mixing it until it becomes colorless. So now we've prepared the sodium bicarbonate for the experiment. Next, we take the Vanish crystal bowl cleaner, which is a blue white powder, and you weigh out about a half a gram of the crystal bowl cleaner because it's a solid material. And we're going to add it to about 5 milliliters of water to make a solution of the Vanish. So we add the Vanish powder. And the 5 milliliters of water. It does take a little bit of time to dissolve the Vanish into the water, but it looks something like this when it's dissolved. Now we're ready to add our reagents. So make sure you take the lid off the cell wall plate. That's always important. So what we're going to show for you today is we're going to show you the different household chemicals with green food coloring and the different reagents with Drano. So right now she's putting about 10 drops of each household chemical into the cell wall plate to see if there's any reaction that occurs. So anywhere from 5 to 10 drops of your household chemical is put into the cell wall plate. So she's taking each household chemical, adding it to the cell wall plate. So now all the household reagents have been added to the cell wall plate. Now we're going to add green food coloring to each cell well to see what happens. So we're adding, somewhere between around 5 to 10 drops of the reagent. And bear in mind that it may take a while for a reaction to occur. So right now it looks like no reaction has occurred so far. We're going to add a few more just to check. And you're always add a few more drops of your reagent. To see if any reaction occurs. So now we're adding a Drano and showing you how each reagent reacts with the Drano. So again, 5 to 10 drops of Drano. Disregard the stuff you see in the lower lower left-hand corner. We had a fiasco with the green food coloring experiment. So now we're going to add calcium chloride, I believe. So first we're going to add blue food coloring, then silver nitrate, then Thymol Blue, calcium chloride, and then of course HCl. Next we're adding the silver nitrate. And so you see with the silver nitrates, when we added it to the Drano, we get a brown precipitate. So remember with Drano, we said that sodium hydroxide was the key ingredient. So if we have silver nitrate, and then we add it to sodium hydroxide, this is going to be a double displacement reaction. And so we get silver hydroxide and sodium nitrate. And remember in the previous slide when we talked about solubility rules, I mentioned that all nitrates were soluble. So sodium nitrate is going to be aqueous. That means that our precipitate is most likely silver hydroxide. So this is how you would explain your results. You're saying, well, when we add the silver nitrate, it reacts with the sodium hydroxide to form silver hydroxide and sodium nitrate, and based on the solubility rules. So that's the reaction that's occurring in the second cell well plate. Also notice that the blue in the blue food coloring is starting to slightly dissipate. So it's reacting with the blue food coloring as well. Next, we're going to add the Thymol Blue, which is an acid base indicator. So when it's in an acidic solution, it's one color. If it's in a basic solution, it has a different color. So you should look up the acid base indicator Thymol Blue to see what color it is in an acidic and basic solution. So here, when we add a few drops of Thymol Blue, since the key ingredient in Drano is sodium hydroxide, very basic, it turns a blue color. Again, disregard what you see in the lower left-hand side. So far, we've seen 3 reactions with the Drano. Next, we're going to add calcium chloride. And you can see here that the calcium chloride forms a cloudy white solution. So again, if we go back and look and see why is calcium chloride reacting with sodium hydroxide, we write our reactants. So we have calcium chloride, we have sodium hydroxide. So we're going to do a double displacement. So we're going to switch the cations and make 2 new compounds. Now, what you should know is that all group 1 salts are soluble, so sodium chloride is going to be aqueous. And if you check your solubility rules on the previous slide, you'll see that calcium hydroxide is a precipitate based on the solubility rules we just showed you. So that's what our precipitate is. It's calcium hydroxide precipitating out. And then next, we're going to add the 6 molar HCl and see what happens to the Drano. Remember, Drano is very basic. HCl is acidic. So we have an acid-base reaction occurring. These are actually pretty exothermic reactions. So if you were to maybe use a thermometer or something and measure the temperature, you might notice that the temperature has increased. Now, one key thing to remember when working with 6 molar HCl is that when you use 6 molar HCl with the Clorox bleach, this produces small quantities of chlorine gas. So make sure you do this in the hood. So go to the hood and make sure you do this part of the experiment. So do this in the hood. So whenever you're reacting the bleach with the hydrochloric acid, make sure you do this experiment part in the hood. It takes like a minute or 2. You add your 6 molar HCl to the Clorox bleach. Give it a few minutes, observe any reactions. And then once you see that if any bubbles have formed, and then you can move it back to your bench. But this part should be done in the hood because when you mix HCl and Clorox bleach, the HCl reacts with the sodium hypochlorite and forms tiny amounts of chlorine gas. So make sure you do this step in the hood. And lastly, we're going to add green food coloring to the Drano. Again, disregard the We had an accident with the green food coloring with the household reagents. So here you would just, and again, don't discard your cell wall plates, give it a few minutes. Make sure that you, give it a few minutes. So things that may not appear to be reacting after 5 or 10 minutes may appear to be reacting. So it's always good to wait and check later to make sure if a reaction is occurring or not. So likewise with the green food coloring, I would wait 5-10 minutes, see if the green color goes away when you add the food colorings to your household reagents. So this is how we complete part 1 of experiment 2. Use cell well plates. You're going to have a list of reagents, a list of household chemicals, and you're going to see how the household chemicals react with the different reagents. Also, make sure that when you're mixing the HCl with the Clorox bleach, this is done in the hood. So now we're going to look at part 2 of experiment 2. So before we get to part 2 of experiment 2, just remember we're using 6 molar HCl. It's very corrosive. If you have large amounts of this spill, it's best to use a sodium bicarbonate to neutralize it first and then clean it up. Again, when you're reacting HCl with the Clorox bleach, do this in the hood, because as you see here, when you react HCl with sodium hypochlorite, chlorine gas is produced. So you will produce tiny amounts of chlorine gas. So this is done in the hood. So not on your lab bench, in the chemical hood. So that's where you should do this part of the experiment. Now in part 2 of experiment 2, what we're looking at is the reactivity of sodium bicarbonate. And so we're going to be reacting sodium bicarbonate with HCl. We'll be using the acid-base indicator, methyl orange. So you need to develop a procedure for doing this. This will be very similar to what you did in the burette lab. So the first step would be to dissolve your sodium bicarbonate in an Erlenmeyer flask before you start the titration with a HCl. And so your TA or your teaching assistant is going to assign you a certain mass of sodium bicarbonate to use. You need to determine the amount of HCl that's required to reach the equivalence point. So this is when the solution changes color, which is the end point. So once you see the end point, this will relate back to the equivalence point. So this is called the end point, when the indicator changes color. So you're going to see how much HCl it takes to get to the end point, and then you're going to share your results with other groups so that you get a variety of masses of sodium bicarbonate and see how the volume of HCl changes with the mass of sodium bicarbonate used. And then you're going to plot the volume of HCl versus the mass of sodium bicarbonate, making sure that you use the XY scatter plot with markers only, and then perform linear regression to see if there's a linear relationship between the amount of sodium bicarbonate used and the volume of HCl used. So again, you'll be plotting using a XY scatter plot, markers only, what you've been using so far. And this is part 2. So I'm going to show you this next, how to do part 2 of experiment 2. So the first thing you're going to do is, once you get your mass of sodium bicarbonate, of course, you're going to come and weigh out your amount of sodium bicarbonate. Make sure you tear the balance. Sometimes you may need to zero. Add in your measure out the amount of sodium bicarbonate given to you by your TA. So for this example, we're going to be using 0.6 grams of sodium bicarbonate. Then you're going to add it to a 125 milliliter Erlenmeyer flask. And dissolve it in 15 milliliters of water. So now we're going to add 15 milliliters of DI water to the Erlenmeyer flask. And make sure all the sodium bicarbonate completely dissolves before you move on to the next step. So now it's dissolved. We're going to add the methyl orange indicator. Just a couple of drops is all that is needed. So you see here, we only added 3 drops. The initial color of the solution is like a yellow orange. I would say more orange color solution. Now we're going to see how much HCl is needed. So we're starting at 0.4 milliliters. We're going to see how much hydrochloric acid is needed to change the color of the solution. So when the solution changes color, that's the end point, which also signifies the end, which also correlates to the equivalence point of the acid-base reaction. So keep on adding HCl until it changes color. Notice how the one hand grasps the stopcock, the other one has the Erlenmeyer. So you're gently adding the hydrochloric acid to the Erlenmeyer flask. So you keep on adding the HCl, and eventually it'll turn this nice peachy pink color that signifies the end point, and that's when you stop adding HCl. So here it took about 16.75 milliliters. So we started at 0.4, it took 16.75 for the color of the endpoint to be reached. The endpoint correlates to the equivalence point, which means that all of the base has been neutralized by the acid. And so when you get your value of the volume of HCl, you'll write it on the board, and other groups will write their values. So then you can plot volume of HCl versus mass of sodium bicarbonate. And that's how you perform the second part of lab 2. So that concludes this pre-lab lecture over experiment 2. I hope you were able to gather some useful information from it. And until next time, Dr. Armand signing off.